Hello everyone, I'm pleased today to present my research project at EACR Goodbye Flat Biology 2021. My name is Carla Faria. I'm a PhD student in Team 9 of CRCT in Toulouse, France, led by Camille Laurent, and I'm under the supervision of Christine Bezombes. I will present our work on patient-derived lymphoma spheroid in personalized medicine, a new model to test immunotherapies. First of all, I would like to present the pathology we are working on, the follicular lymphoma. It is the second most common type of non-Hodgkin lymphoma. This cancer is composed of malignant B cells derived from germinal center. FL usually has an indolent course and excellent overall survival. However, the disease remains incurable with conventional approaches and is characterized by repeated relapses. Thus, FL research focuses on the development of new and more efficient molecules. To this end, the development of relevant in vitro models is crucial for testing new drugs in preclinical settings. Here, we present a simple workflow to generate patient-derived lymphoma spheroid established from dissociated FL biopsies adapted for high-throughput model imaging and drug screening. This work is not yet published, so we are keeping confidential some key points about the methods. Here's an overview of our workflow. We cultured whole lymph node cells from follicular lymphoma patients with an enriched medium in round-bottom 96-well plates in ULA ultra-low attachment plates. Those models are called PDLAs for patient-derived lymphoma spheroid. After 3 days of culture, we added the drugs to be tested, an anti-CD20 obinutuzumab in combination or not with an anti-PD-1 nivolumab. After 3 days of treatment, we evaluated morphological features and cell growth behavior by 2D and 3D imaging and flow cytometry in order to assess the robustness of the model and treatment efficacy. First of all, we characterized the model in depth thanks to 2D and 3D imaging. On the bright field images that you can see on the panel A, we can observe an interpatient variability in the 3D structuration with a model composed of 2 parts, a monolayer and a 3D part. This observation was confirmed thanks to 3D images presented in the panel B. We also performed immunohistochemistry and immunofluorescent microscopy in order to visualize B and T cells localization. We can observe that the majority of the model is composed of B cells and some clusters of T cells are homogeneously distributed in the model. Then we performed a cell counting along the days of culture and we assessed a drop between the day of seeding and 3 days after 3D culture, followed by a stable cell number until day 6 of 3D culture. Thanks to flow cytometry, we continued the in-depth characterization in the immune cell distribution and we observed a high percentage of CD90 positive cells with around 60% of total cells, and the rest corresponds to T cells with around 20% of CD4 T cells and 10% of CD8 T cells. We also observed the presence of few gamma delta T cells and NK cells. Next, we went further into T cell characterization with the immune checkpoint expression of BTLA4, TIGIT, LAG3, PD-1, TIM3, CD39, and CD73. For CD4 T cells, we observed a high percentage of cells expressing BTLA4, TIGIT, PD-1, and CD39. The same goes for CD8 T cells, but with the addition of the CD73. The next step was to evaluate the efficacy of immunotherapies on PDLAs treated at day 3 of culture during 3 days. This efficacy was determined by imaging to evaluate the effect of the treatment on the morphology, and then by flow cytometry we determined the B cells depletion. In bright field images and in 3D images, we observed that the anti-CD20 have the tendency to compact the model. We were able to assess an increase in destructuration and a decrease of the model area with all the drugs with an interpatient variability. To finish, we monitored B-cell depletion thanks to flow cytometry, and we observed a B-cell depletion with almost all the treated PDLS, but we observed no increase in B-cell depletion following treatment combination. In conclusion, this 3D model exhibits characteristics similar to FL biopsies. By 2D and 3D imaging, we were able to analyze their morphology and the 3D distribution of proliferation and cell death in T cells and B cells. Detailed characterization of PDLS composition by multiparametric flow cytometry analysis showed an interpatient variability with potent differences in the BT ratio. Moreover, we determined for each PDLS the expression of immune checkpoint on T cells. Finally, we tested drug screening by using anti-CD20 and anti-PD-1 antibodies combination, and we evaluated the effect on PDLS morphology, but also on lymphoma cell death. This is the first 3D FL model exempt of matrix or other compound used to keep aggregated tumor cells relevant to the pathology with the same pattern of histology. We believe that this model presents strong evidence to become an interesting preclinical model for personalized medicine. I thank you for your attention, and I would like to thank all our collaborators. Please contact me if you have any questions.